much. Yeah, welcome to this workshop on AI and health. I just want to start by saying a big thank you to Pat, Sarah, and Nina for organizing this. It's been a real pleasure to work with them because they've essentially done everything for us. So I don't need to tell you, this is a really exceptionally exciting time to be working in AI, and specifically AI with health. We've seen some really massive advances in the tools that are available to us. So just from a slightly selfish perspective, I'm a protein designer. Just focusing in on that for a second. Some of the tools that have come available over the past couple of years, so we're talking about AlphaFold and the tools that have come from the Baker Lab, have really surpassed what we imagined possible, right? They're making something that was incredibly complex in the past actually relatively routine now. So what this allows us to do really is to take on ever greater challenges, and hopefully we'll see the impact coming into the sort of health sphere from this. We also have the slight benefit, and this kind of came up in conversations between Nina, Sarah, myself, and the other organizers, that we have the sort of benefit of not having really the stigma of a possible stigma of AI. Ethically speaking, AI for health, people are generally very on board, I think, with us using these types of techniques to improve healthcare. So today, this workshop really is about providing us with a really timely, unique opportunity to hear about some of the activities across both the university and the NHS, and allows us to look for connections and synergy with our research. And as Nina said, there's ample time for us to talk about this afterwards in the networking session. We should also think about finding new ways to really start exploiting the amazing capability we have in Bristol through things like Isambard AI and the Doctoral Training Centre in Engineering Biology. So that's really it from an introduction for me. I am going to now introduce our first speaker. Are we queued up for that? Is that right? So, yeah. So real pleasure to introduce our first speaker. So Dec Wolfson from the School of Chemistry, Bristol. Dec has been a protein designer for his entire career. In fact, yesterday, Dec was awarded a Fellowship of the Royal Society. So big congratulations to Dec. And just to say that with the power of AI and what it's done for protein design, even Dec, who is a rational protein designer, has started to use AI in his research. It was also suggested, by the way, that this is your inaugural talk. Daryl suggested that you should consider this your inaugural talk for your FRS. Anyway, could I invite you to? Yes.
Shall I get going? So most of you know me, some of you don't. Those that do know me know that I get carried away, so I'm going to get the timer going for 18 minutes, wasn't it?
10.
I'm going to set it for 8. Okay. And then I'll just make sure I can wrap up to that. So we've got a pointer. Cheap seats, can you hear me okay?
Sorry.
I'm going to stand over here. Oh shit, I don't know. Really cramps my style. Okay, I will try. Yeah, it's okay, I'll try. So a few things just to get started. It's a real honour to be kicking this off and to have the luxury of so much time to talk. And I'll try not to abuse that and be on time. Also, I think it's a really exciting time for all the reasons that Ross has just said. And I think it's also a really exciting time for another area that this impinges on, that's synthetic biology. And I can't give too much away, but I know there's now a new Jedi Council at the university in synthetic biology, and I'm really excited to see what happens there in this sphere. The fun thing I want to say, and this is as just as an introduction, is that despite all these advances, I hope you're with me in that we don't understand anything, right, about proteins. And AI just gives us those tools to get into places that we really don't understand. And I'm hoping that we'll have something like an explainable AI in years to come that will actually combine what the kind of stuff I love as a physical scientist, as a chemist and a physical biochemist, and the power of AI. Because I kind of think without that we're a bit lost, to be absolutely honest. I think we need this relationship between chemical sequence structure and ultimately function. My backdrop here it looks pretty impressive, right? These are all X-ray crystal structures of de novo designed proteins. None of them are built with AI. They're all built rationally or they're built computationally. All right, that's just exploded. We now have hundreds of protein structures because of all of these methods plus AI. So I thought I'd better just set the scene. Protein folding is the forward reaction, taking a protein sequence, so a linear stretch of amino acids, folding up into a beautiful structure. Protein engineering is the idea that if you change one or a small number of amino acids in that natural sequence, you're doing a protein engineering experiment. That's part of the forward protein folding reaction. The reverse of that, where you take a protein structure or a protein function and you want to generate or design a protein sequence that will fold to that, is the protein design problem or the inverse protein folding problem. Is everyone kind of with me? And of course, the states of the art of these things, AlphaFold and others going forward. The reverse is now, as Ross introduced, generative AI. And this won the Nobel Prize. These two things won the Nobel Prize. So David Bacon, protein design, and Demis and John for AlphaFold. Why do I do it? I've already given this away. I want to understand sequence structure relationships, so I can go in there with like really scalpel-like precision and make changes to de novo proteins to make them functional. But what other people are doing, of course, Ross does beautiful work in this. This is trying to make a catalytic protein. This isn't Ross's work. This is actually a Baker design from Computational Design with Berta Herka. It's 10% of all the enzymes have this structure. If you don't know it, it's called the tin barrel. 10% of all the enzymes have this structure. That means it's a privileged protein structure. My understanding is, from Don Hilvert, who's probably one of the best protein engineers on the planet, he can't evolve that into a functional enzyme. So even though we can build this as de novo designers, Ross might correct me on this, people haven't really succeeded in changing this particular scaffold, a privileged scaffold, into a catalyst, right? That's worrisome. This is something we built. It's a switch between two states, an open and closed state of a helical bundle. You can use those things to sense metabolites, things for biomedical sensing, for example. This is a protein in colour from Bruno Correa's group, where he can make a picomolar, really tight binder, better affinities than antibodies, to a protein from a virus that he wants to neutralise, right? So that's the hope. The way people do this, sort of been introduced minimally, that's just using very simple chemical principles, separate hydrophobic and polar residues. Rationally, that's embellishing those with our bioinformatic and chemical understanding of proteins to put those into designs computationally, so we can look at all manner of sequences that might fold up into the protein that we want. And now with AI. Just to put this into perspective, this is how we do things. So we use chemistry-based or physics-based rules to build helical assemblies. So we can instruct small polypeptide chains to fold up to find 2 copies of themselves, 3 copies of themselves, and so on, all manner of combinations of those things to produce discrete protein objects. And that's just using pen and paper in biro design, if you like. We can do that because we have lots of examples of this in the literature or in nature. Where we don't have so many examples, these are called alpha helical barrels. We just discovered them by chance, by messing around, right? And we found some proteins with holes in them. There are very, very few examples of these in the natural sequences and structures to learn from. So we had to build our own physics-based computational methods to build these things. And then what Ross alluded to again is that we can use this understanding that we built from rational or computational things, and we can now use AI to join those things up. Now, all of those things on the top 2 lines, they are small proteins called peptides that we build chemically with a peptide synthesizer. These things on the bottom are real proteins. We make a synthetic gene and we express that in E. coli, and they fold as we predicted them to fold, right? But we use the things on the top as seeds for our computational design, and this is aided and abetted by AI. That stuff on the top took my group, really talented people, some of them sitting here actually, 15 years to do, right? Then AI came along and we were able to take those rules that we learned and make these proteins in less than 5 constructs. We make no more than 5 genes. And we always get a crystal structure, and they look like what we predicted in the computer. What are we doing with these things now? Ross does similar things, other people do similar things, Paul does. We've built very, very simple physics-based methods, not AI-based methods, although we use AI in the pipeline, to build holes in proteins that will bind small molecules. And we've bound natural flavin molecule. This is a one-shot design. One design, one gene, it binds our target flavin with micromolar affinity. I will pass over this because I guess that Jess might talk about this. But essentially, we can also build proteins that will take things into cells and hijack processes in cells. In this case, we're hijacking a molecular motor, and we can get the design protein to carry things through the cell on the molecular motor. We can also build materials inside cells. So these are liquid-liquid phase separated systems or biomolecular condensates, very trendy, bit weird in biology. But we can build these de novo, and they're collections of de novo proteins to which we can sponge in de novo chemical functions. That's 8 minutes, so I've still got 2. Is that okay? Yeah, right. But I'm pretty much finished there, right? So here are some questions and some conclusions that we can maybe talk about later on. We can maybe put this up later. Just my indulgence as being as the keynote to leave you with those. But what I wanted to tell you about in 1 minute is that I've just been asked to start a new Centre for Protein Design in Copenhagen. And this isn't going to be introspective. This is going to be broad and open. And Bristol is going to be one of the spokes for this. So we're going to have 3 themes. I've already outlined those. How can we program de novo design fully? How can we do protein design in the context of a cell or a reaction? And then how can we use protein design in health and sustainability? As I say, we're going to reach out. We're already reaching out across Europe. Bristol should be on there. Oh, yeah, it's on there in a minute. We're reaching out to UCSF. That's going to be one of our spokes. And Bristol's going to be one of our spokes. So there are going to be real opportunities for people to dip into this centre from Bristol. And that's about all I want to say. And we're going to have mechanisms for collaboration. But that's about it. Thank you.
Thank you, Jeff. And our next speaker is Lucia.
Yeah, thanks everyone for the opportunity to present today. I'm going to very briefly introduce you the research in my group. Is an interdisciplinary group composed of both experimentalists and computational researchers. And the overarching aim is to try to make the engineering of living systems more predictable and robust. And to do this, we mainly use 2 methodologies, modeling and AI and feedback control. Let's start with modeling. So modeling has been used since early days in synthetic biology. At the start, mainly to design and tune gene networks that you can see here. They were simplified systems with a few genes regulating each other. For sure, biology is much more complex, and in the cells there are many more interconnected processes. And that's why we now move to the next generation of mathematical models in systems and engineering biology. They are called whole cell models. They represent cells at the genome scale levels, and they aim at representing all the cellular processes known in a cell, for example, DNA replication, transcription, interactions among proteins, and metabolic interactions as well. And importantly, these models can predict phenotypes from genotypes. This is why in the past few years, working closely with Claire Gerson, we have been trying to understand if and how we can embed these whole cell models and AI into DBTL cycles. Why? Because having a digital twin of a cell should make the redesign of entire genomes much more predictable and fast. So we are trying to embed these models into the design and learn phases. And for sure, we are also using AI, both to reduce the computational burden of simulating these models, because they're composed of thousands of equations, so we need to reduce the time to simulate them. We also need smart ways to analyze the outcome of these models, and finally to compare in the learn phase the outcome of these models with the experiments that we're performing. So far, we have used both existing whole cell models. One is for Mycoplasma genitalium and the other one for E. coli. In terms of the applications, we mainly focused on genome minimization, and we have designed thousands of genomes in silico. We are now going to test some of these in the lab. So we just got a grant. We're working with Tom Gorochowski here. We are going to build some of these reduced E. coli cells, and we are going to test them as chassis for metabolic engineering. What about feedback control? This is another approach that we are testing in the group. So this is a concept that we borrowed from engineering, where feedback control is broadly used to stabilize systems. And this can be applied to biology. We are doing this in different ways. We are building controller into cells, but also we are developing platforms that allow to control cells externally. For example, in that video, we are trying to steal a population of E. coli cells to steer and maintain the SAR levels of a GFP protein. How we do this? We have developed a platform where we culture bacterial or mammalian cells in microfluidics. We do time-lapse imaging. We apply different imaging segmentation tools, also based on AI, to quantify the output of the cells. And finally, we close the loop. So the AI is both here in the analysis of the images and here in the control algorithm. We developed this platform again for different types of bacterial and mammalian cells, and we are also now trying to apply this platform not just to control a GFP, but also to control other phenotypes. For example, there is an ongoing collaboration with AstraZeneca, where we are trying to exploit the control algorithm to predict combination therapies for cancer, to avoid or at least delay the emergence of resistance to drugs. And also we are trying to use the platform to prototype in a completely data-based way, so without the need of models, gene networks in both bacterial and mammalian cells. That's it from me. I just wanted to mention that I'm also directing the CDT in engineering biology that Ross mentioned. So if anyone in the room is not working with us but would like to engage with us or possibly to supervise students, please do get in touch, because there is the possibility to do that. Thank you.
Daniel next.
Some other slide.
That's brilliant, yeah. We took note of that.
Thank you, Nina. Good morning to everyone. My name is Daniel D'Andrea. I'm a senior lecturer in data science in the School of Engineering, Mathematics and Technologies. This is a slide just to summarize my research interest. Basically, what I try to do is to integrate different type of data, in general omics data like transcriptomics, genomics, metabolomics, along with the clinical information, and applying a different type of approaches like statistics, bioinformatics in particular, but also machine learning and AI. I try to identify some mechanism affecting complex disease, or try to identify some new methods to be able to analyze this type of complex data, in particular to support researchers to use this data, but also try to identify new experimental directions, in particular for the area of precision medicine. It is what I have done in the last 15 years, and in particular I have applied this type of approach to different type of disease, in particular cancer, but also psychiatric disorders and rare disease. For instance, now we have, in collaboration with the Imperial College, a clinical trial for the first-in-class inhibitor for multiple myeloma and diffuse large B-cell lymphoma. Today I would like to speak for a new project that I'm doing, basically I started last year. It is about cancer. When we speak about cancer, you know that we have cancer cells, but the cancer in general is something that it is a very complex environment. So we can have different type of cells with interaction, and this tumor microenvironment is associated with the tumorogenesis and cancer progression. We decided to focus on a specific agglomerate of immunocells that are the TLS, tertiary lymphoid structures, that emerged as a prognostic and predictive biomarker of response to immunotherapy. So by using different type of data, starting from clinical information that we can have for patients to have a snapshot about the tumor's composition and key features, we decided to use also omics technologies, the standard one, that are the bulk sequencing, so analyzing the entire tissue of cancer, but also the single cell sequencing, so we are able to identify the materials in the any specific cells. and also spatial transcriptomics, where we can also identify where the cells are in the tissue. Obviously, individually, these technologies are not able to identify and characterize well the tumor microenvironment, but maybe together we can have a good understanding about what's happening in the tumor microenvironment, and in particular, in the TLS. So what we started to do in collaboration with the Italian Institute of Cancer, I work with them just because they already have some patients where they characterized our lung cancer patients, so characterizing poor, good, super good. They have blood, adjacent on tumor sites and also tumor sites, and perform an experiment to have different type of omics, spatial transcriptomics, but also single cell omics. We are able to identify this TLS in the tissue and try to identify some dynamics that are able to be associated with the response to immunotherapy and tumor progression. So as you can see, this type of research is a multidisciplinary research. So what I'm looking for, and the input and expertise that I'm looking for, is something that it is related to oncology, medicine, immunology, not only to have patients or experiments, but also some potential new hypotheses that we can test on public available dataset, but also data scientists to identify some new methods to analyze this complex system. That's it. Thank you.
So we are now moving to our lightning talks presentations, and just we have a new speaker on the program who doesn't appear on your printed programs. It's a late edition, so very exciting. Yeah, so it's Liam Fowler, and you will present yourself.
Yes, so
the program goes as per printed program.
Yep. I'm Liam Fowler. I'm in the School of Biochemistry. I started about 3 weeks ago. And we're interested in ribosomes. Ribosomes are incredibly abundant, about 10 million, between 1 and 10 million copies per cell. And only recently we've realized that not every ribosome is the same, and that there's variability and that there's actually specialization of ribosomes. But at this stage, we've got lots of questions in this field, like how common these are, the function in health and disease, and even what defines it. And one of the big issues is that there aren't good tools available to answer these questions. Now, we've developed an approach where we can use ribosome profiling, which is an information-rich technology to look at the translation that's going on in a cell. And we can use this information to get to look at the ribosomes. And the real advantage of our approach is that we can look at previously published data. So there are thousands of studies that have been published using ribosome profiling that we can now go back and look at the presence of these different ribosome populations in that data. So we can take all of the data from that ribosome profiling, things like the translation efficiency, gene set enrichment, stalls, collisions, huge amount of data, and use these as features in a machine learning approach to begin to answer some of these questions that we have and understand how important this ribosome specialization is.
Thank you, Liam. Now we have John Lees.
Hi, so yeah, just a minute, so I won't talk about the Man United match last night. Unfortunately, a Man United fan, so there we go. But yeah, I'm working bioinformatics. I'm down at the medical school. Over my career, I've sort of worked on lots of different areas of bioinformatics, but since moving to the medical school, we've been increasingly working on medically related questions. So looking at things like cell therapies for cancer, using AI to help improve those. And since Isambard's been here, we've been using that increasingly as well for our research. So over a number of things we've been using, we've developed these things called in silico pull downs, where we are able to take a protein of interest and pull down against the rest of the genome. So around about something like 1 million interactions we predicted with that now. But also just we're kind of very highly collaborative. So just to highlight another example where we've used Isambard AI, is we modeled this beast of a biomolecular machine called the spliceosome. And basically we took mutation data and we're able to give a mechanistic interpretation for the mutations. So able to basically patch up why the mutations affected the spliceosome and were driving this disease that was related to neurodevelopmental disorders. So that's a very quick overview.
Thank you, John. Thank you, John. And now we have Darryl. No, it doesn't. Oh, there we go.
Hi everyone, I'm Daryl. So we work in infectious diseases and we've designed a very rapid point of need diagnostic based on gold nanoparticles conjugated to DNA. And these target pretty much any bacterial cell you want them to and can be tuned effectively. And this is based largely on highly repetitive stretches of DNA copies that are present thousands of times in the genome. That's great if you're looking for a single species. We're using, with Iril, who's here, a relatively straightforward algorithm, but what we need to do is make sure these are strain, species, genus specific in complex scenarios, and that's where we think AI can help us. And rather than being a very visual, and there should be a nice little thing at the bottom there showing you the presence of, maybe ever do that, yep, presence of what the test looks like, it's a yes, no, but I think with AI we'll be an really interrogate that surface chemistry, the density, all sorts of deep tech approaches to developing these so they will be quantitative, not just qualitative. Thank you very much.
Thank you, Daryl. And next up we have Karen Lowe.
So complete change of tack now. I'm a clinician, one of the very few women in the audience as well, so bear with me. Essentially, this is about the general cohort study, which I've been chief investigator of in the UK. It's 547 children with monogenic neurodevelopmental disorders. We've got deep phenotypic data, which has come from both parents, clinicians, and we also have genomic variant data. So it's probably representing one of the most detailed phenotypic data sets in this sort of rare disease to date. And essentially, we have lots of ideas about machine learning applications. Some very blue sky ones, which are possibly much harder to achieve, but also some much smaller projects, which includes one which I'd really like to be able to do, which is a way of using AI to help semi-automate some of the research processes within the cohort study. So, for example, I've got a very talented junior doctor working on Tub A1A at the moment. She's doing some lovely work synthesizing all the data that we have. And what I'd like to be able to do that is to do that for all of the genes in GenRock. But it's a lot of work manually, and so it's whether we can find a way to replicate that process that she's been doing. I'm also working with Edwin Simpson on a couple of small NLP projects, looking at converting the free text responses from parents into HPO terms, and also looking at using literature to generate reports for the study participants, and I'm done. Thank you.
Thank you, Karen. And Jordi Paps.
Hi. My name is Jordi Paps. I'm from the School of Biological Sciences. A few of you might know me because I used to be the director of the Master in Bioinformatics, and I was chasing you to take research project students, but I quit that. I hope you can forgive me. My area of research is comparative genomics. I'm very interested in understanding how genome changes have driven measured evolutionary transitions. So, for example, in my research has focused on the origin of the animal kingdom, the origin of land plants, and stuff like that. However, I'm also interested in parasites in the animal kingdom. So this is a phylogeny of all animals, an evolutionary tree of all animals, and the ones with stars are parasitic. So we have a pipeline in the lab in which, using an evolutionary framework, we can reconstruct ancestral genomes, so we can tell all the genes that were present in the very first parasitic flatworm or parasitic nematode, etc. And then we can pinpoint which genes are unique to this ancestor. So we can find genes only found in parasites or only found in all parasites. So I feel kind of an intruder because I'm not doing biomedical research. Actually, I don't do AI. But in the future, I hope, we have this pipeline, and then we can characterize the functions of the genes in those genomes. So in the future, I would like to use AI to try to predict which genes we expect to find in a parasite. And I would also like to find friends, so they can help me to understand how those genes in parasites interact with the host, and also to design drugs to fight those parasites. Thank you.
Thank you, Jordi. And next we have Dan Lawson.
So hi, I'm interested in methods for AI as well as applications of population genomics, and I was you've probably been bothered by me about this DTP I was applying for, AI for cross-cell biosciences. You'll hear more if that's successful. So my project in that is about hybridization. So we have lots of observations of hybridized species, and we see which bits of the genome we see over represented in one species compared to another when they hybridize. And we have this interesting pattern that everywhere we've looked so far, we find that immune genes stick out like a sore thumb. And this is interesting intrinsically, but it's potentially useful as well if you're interested in conservation and understanding differences between species that are hybridizing, either the ones that do this by accident or ones we're doing so on purpose through breeding programs in conservation or in agriculture. So, we'll be definitely using AI to figure out the function of that, and that's what the project will be. All right, thanks very much.
Thank you, Dan. And now flipping over the sheet, we're back to Ross.
Thanks to me. Okay, so my group works on what we call the de novo design of what we call bioelectronic proteins. The reason we do this is to better understand some of the really incredible processes that happen during cellular respiration and photosynthesis. So we build proteins for electron transfer, right, as a means of understanding this process. So these are some examples we have at the end there. The way we do this, so we use AI in some of the design methodologies. We're very much end users in this. To study these proteins, we use various types of spectroscopy, so ultrafast spectroscopy, looking at processes that happen down to the femtosecond. We also have been more recently doing single molecule work, looking at conductivity through these wires at the single molecule level. And that's it. Thank you.
Thanks, Ross. And now we have Paul Curnow.
Thank you. Afternoon, everybody. So my lab are also a protein design lab. In fact, I share a lab with Ross. Our interest really is in integral membrane proteins. So that is proteins that sit inside a membrane lipid bilayer. They're a bit understudied in the design world, not because they're boring, but because they're more challenging to study. So we're now using the same design tools that you've heard about already to design, ideally, functional membrane proteins, specifically for electron transport across and within membranes. The second thing that my lab are trying to do now is to develop sequence to expression models for membrane proteins. You may not know, but the recombinant production of membrane proteins remains probably one of the most outstanding challenges in protein science. It turns out computationally designed proteins are an excellent basis for building models that can link a particular sequence to a particular expression phenotype. So if you're interested in membrane proteins, come and talk to me.
And following on from Paul, we have James Armstrong.
I'm not sure James is here because his seat is...
No, he sneaked in at the back. I spotted him.
I sat in the wrong seat. I'm James. I am a senior research fellow at Bristol Medical School. What we're very interested in is growing tissues, a whole host of different tissues, and using these as platforms to understand how different genes or drugs affect these tissues. So it's healthcare applications. And AI or machine learning, we're very interested here in taking these quite large input data sets and using these to better understand and predict these different, very complex biological responses. And this is just an example of a sort of project here. So this was partly EBI funded. I can't actually show this. But what we did here is we used an AI framework to analyze and classify the development of these very complex brain tissues known as organoids. And very excitingly, we're able now to predict some of those developmental trajectories at very early time points. So potentially a very useful toolkit here for researchers in the field. And that's all. Thank you.
Thank you, James. And now we have Jessica Ross.
Thanks. Hi, I'm Jess. I'm a new lecturer linked to the Engineering Biology CDT. I did my training with Dec Wilson at learning how to design and understand core domains, and with Mark Dodding in biochemistry learning how motor proteins move things around inside cells. And what I'm really interested in doing now is using protein design and engineering to understand how these motor proteins go wrong in human diseases like neurodegeneration and Alzheimer's and ALS, and how we can develop new tools to switch them by designing these switches to manipulate their transport, so to turn them back on when they get dysregulated or to switch them off when they're hyperactivated. And also learning how these proteins work by building them from the bottom up and taking them apart and building de novo domains in order to understand how these motor proteins function and how they're regulated.
Thank you, Jess. And now we have Ben Hardy.
Hi, so this might seem familiar. I'm a postdoc between Paul Kerno and Ross Anderson. So you kind of heard a bit about what our proteins do. We're designing redox proteins. I thought I'd just mention some of the points where we're using AI through design. So we're using AI, we can design protein structure to bind redox active cofactors. We're using AI to design the sequences, predict their structures, to improve these designs iteratively on the computer. But we also take these all the way through to atomistic simulation. We create these proteins, express them in cells, and we can reconstitute our membrane proteins into sort of model respiratory or electron transport processes in liposomes. And I think what would be quite interesting is to see what is the next step from here. So going from the model systems in the labs, now we have robust control over electron transport in membranes. Can we find some sort of medical applications to this, which obviously is beyond our expertise, but we could achieve through some collaboration. So I think if any of this seems interesting to you, or if you want any help maybe using some of these AI tools for design, come and talk to us. Thanks.
Thank you, Ben. And we now have Robin Corey.
Hello. So in my lab, we're interested in using computational modelling to assist the drug discovery process. AI is helping in a number of different ways with this. I'll go through each one of these in turn. Firstly, we're using AlphaFold, of course, as well as other programs like BioEmu to model target structures. So for us, targets are typically membrane proteins. And BioEmu allows us to capture a number of different states, which can be really important for drug design. We're also trying to predict lead target interactions, so how small molecule drugs might interact with membrane proteins. This is using AI improved docking methods, but also just de novo AI modeling, such as with Chai-1, so we can predict how a drug might bind to a protein. And then finally, analyzing complex simulation data. Ignore the one on the left, but on the right, so we generate loads and loads of data, and we can't really analyze it, so we're really interested in getting AI algorithms to help us analyze this. And we've got a collaboration with the Thompson Lab, but also open to discussions on this point for anyone else. Thank you.
Thank you, Robin. And now we have Jim Spencer.
So, good afternoon, everybody. I'm Jim Spencer from the School of Cellular and Molecular Medicine. Most of what I do is related to antimicrobial resistance in bacteria. In this context, I want to talk about efforts to develop novel antibacterials, specifically to understand the mode of action of novel antibacterials, so how they kill bacterial cells. So the approach that we're using is an image analysis-based approach developed by collaborators called bacterial cytological profiling. So originally around 10 years ago, what they did was label bacterial cells, challenge them with antibiotics, and use principal component analysis to classify morphological responses to agents with different modes of killing, and use that to understand killing by unknown agents. That was then. What our collaborators, who do all the heavy lifting, we do the image acquisition, are now doing is extending this to the single cell level using deep learning approaches. So we label bacteria in the presence of different antibacterial agents. We measure around 60 morphological features and use deep learning then to classify these features and compare with our unknown agent here. So what we can see is that our investigational compound is doing things to bacteria that are different to known agents.
Thank you, Jim. And now we have Ben Faber.
Thank you. So hi, I'm Ben. I'm a academic rheumatologist, and I'm interested in osteoarthritis, which is the most common cause of joint pain. Currently, we have no medical cure, no pharmacological cures for this. So what I've been studying over the last 6 years is a huge array of imaging that we have in UK Biobank, and use machine learning to analyse this imaging of the joints. So you can see the hip and knee up there. And what that's left me with is a sample of over 65,000 individuals where we have their genetics and their imaging. And that's what I've been studying, and I've just been awarded a Wellcome Fellowship to keep looking at that. But what I really want from you is, if people have got expertise at machine learning, deep learning, to be able to integrate imaging and all this omic data that UK Biobank have. We've got genetics, proteomics, metabolomics, and the patient factors to improve our understanding of the underlying biology and hopefully discover new treatment targets and also potentially predict progression of this disease, so we can really target interventions at the individuals most likely to progress rapidly, which we currently can't do. So please talk to me at lunch or email me if you're interested in collaborating. Thank you.
Thank you, Ben. And now we're moving on to 3 talks which are more about support for researchers rather than the actual research. So first up is Richard Gillam. Thank you.
Good afternoon, everyone. We've fed the lapsed chemist in me this morning. Thank you very much. I've enjoyed listening to what everyone's been up to. But I'm really here to tell you about Isambard AI, which will be, when fully commissioned, the UK's fastest supercomputer. And we host it in Bristol. And it is operated by the Bristol Centre for Supercomputing, which is part of the university. And Isambard AI comes in 2 phases. Phase 1 is operational right now, and when it came on about a year ago, it was number 128 in the world, and showing its green credentials was second in the so-called Green 500, measuring the energy sustainability of the machine. Over the summer, we will have a 30 times uplift to this, to over 5,000 NVIDIA GPUs, so we're talking an unprecedented amount of compute that's available to the UK, but also to Bristol University. And we've seen loads of good use cases, some of which we've already heard today. So we've heard with large language model fine-tuning, drug discovery, protein design, we've had AlphaFold running, medical imaging as well, all examples of things that we've done. Have a look at the website to find for details. In terms of access, a very common question we get, there's 2 access routes that applies to University of Bristol researchers for Isambard AI. First of all, the main allocation of time on the machine comes through the DSIT expression of interest, not through the usual UK RI approach. It's funded differently, so that's worth noting. But there's also University of Bristol allocation, which is actually very, very large in comparison to supercomputers that we're used to in the UK. And what I'd like to emphasise is, in terms of user experience, we're trying to democratise supercomputing. So it's not just for the astrophysicists, for the people doing quantum mechanics and molecular dynamics folks. We're trying to be aware that different researchers have different skill sets. So we're trying to be as careful as you can about how we design, how you can work on a system. We're also aware that you already have software designed working on other machines. So we're there to respect your software stack choices, so you bring what you have with you, get it up and running, and get things done as well. And we're acutely aware, particularly in the medical arena, that data sensitivity may be important. We have some users that need to work at government official, loads of users that may have GDPR considerations, and we're just coming to the end of our CAF compliance process, which is the first step of cybersecurity certifications that we want to establish on the platform. Thank you very much.
Thank you, Richard. And now we have Paola.
Wait, I think there's animation here. Apologies for all HDAD neurodiverse audience. These slides are a bit overcrowded, overwhelming. I'm just here to talk about what we can offer from the Jane Golding Institute. Starting from the bottom, because it's the area I lead, we offer courses in different aspects of data science and software engineering, as well a bit of focus, especially this last year, on showing best practices and how to make your research more reproducible. These courses are, most of them, designed to offer to the different doctoral programs, but we are also offering for free to all university staff and students. And if you have in mind something that you want to get some training on, but you don't have people with this expertise, or you want some support, contact us. We always have time to discuss about possibilities to develop new training. We can also offer support in your data science projects, from using the free help desk Ask JGI that is run by PhD students, that they can offer you up to 8 hours of free support. And you can, if you need, you expect that you need more support before writing your project or during your project if you have already started, you can contact us and try to seek for support of a data scientist. We can talk about costing. In the best cases, we can offer up to 6 months of 50% time of a data scientist. If you are looking for the short term, probably we will not have the capacity to offer that, but we will try to adapt the best as possible. Lots of stuff there. I think that important is that you have the webpage there of the Jim Golding Institute. You have all the resources there. And just going to the last slide, we have a lot of networking events and what we recently call interest groups. Particularly, I like to highlight Health Data Research Network. We are running an event on the 9th of July that will try to connect cardiologists with people more expertise in data science. If you are interested, please write me an email. And as well, we can help you in terms of preparing your finding if you want to have some letters of support or a bit of guidance on the part of methods and data analysis. And that's it. Thanks very much.
Thank you, Pau. And our last speaker is Harriet.
Thanks very much. It's really great to be here and really enjoy going back to my roots and hearing a little bit more about protein design. I wanted to talk a little bit about the support that we can give you at DRE to take your research out of academia and have impact in other areas. I'm from research commercialisation, and we work with academics across the university who are looking for commercial outputs of their research. So that could be assessing what markets your research could have impact in. It could be that you think that you need a patent, or you think that maybe you need to protect your work via copyright, for example. We can guide you through that IP protection process. And then we can take you all the way through into taking the intellectual property within your research project out into the world. So that could be licensing to an existing company. It could be helping you to found your own company. So that's kind of what we do in research commercialisation. But we work really closely with some other teams in DRE. Firstly, the impact development team, we work very closely with them, and I wanted to particularly highlight the Translational Research Hub, who are a one-stop shop for health interventions. They can help you all the way from very early stage all the way into taking it out of the university. And we have Susie here who is representing them. So if you want to talk to her, she can offer you some really great support. The other one I wanted to really highlight was the IAA funding that we have. So this is initial prompt priming funding to take your research into that translational space, enabling you to bid for larger external support. And finally, I think that Policy Bristol and public engagement, particularly in AI, are really important to maximising the outputs of your research, making sure that people are aware of and are comfortable with the impact of AI is going to be critical for everyone to benefit from your research. So I'd really encourage you to engage with those teams as well. Thank you very much.
Thank you, Harriet. And now I'm going to hand you back to the three leads of this workshop, who are Daniel D'Andrea, Lucia Marici, and Ross Anderson, just to give some closing remarks. Or it may just be one of you. Yeah. It may just be Daniel.
Yeah. Just to say that it was a very intensive hour, so a lot of talks. But I think that you enjoyed it, because actually for me I had a lot of good and interesting feedback and input. So I would like to say again thank you to Sarah and Nina for organizing everything, because without their support it was impossible. Thank you PATH and ABI for the support. And yes, I think that, and I hope that we can repeat again this event next year, maybe with a bigger audience and with more talks, because I enjoyed it, and I hope that it is the same also for you. Thank you.